Hello, this is Catherine Zhang from DNA Learning Center at Coastman Harbor Lab. This is part 2 for PTC Lab. In PTC Lab, we're going to learn how to use a single nucleotide polymorphism to predict bitter tasteability. That will help us to understand the genetic basis for precision medicine or personalized medicine. Let me recap what we learned from part 1. In part 1, we learned PTC bitter taste ability is a recessive trait, could be passed on from generation to generation following Mendelian's rule. Let's say if dad has all tasting receptors, he can taste PTC. His genotype is big T, big T. If mom has all non-tasting receptors, she cannot taste the PTC, and her genotype is little t, little t. Their children will have both tasting receptors and non-tasting receptor. So they can taste PTC, but their genotype will be big T, little t. We also learned the genes responsible for PTC tasting was identified in 2003, and their polymorphism associated with PTC tasting. To be more specific, nucleotide position 145, if you have C in the DNA sequence, you're more like to be a taster. If you have G, you are more like a non-taster. That's because a single nucleotide change, very small change, at DNA level could lead to a change at amino acid level. Proline changed into alanine. Therefore, the receptor protein can be changed and the function differently. That's why a small change, such as single nucleotide polymorphism, could lead to different ability of bitter tasting. To study this, we need to collect cells, concentrate cells, extract and purify DNA, then use that DNA to do PCR amplification, and later on use restriction analysis to find out the genotype. And we are also going to use PTC paper to test the phenotype. That will help us to understand the relationship between genotype and phenotype. And at part 3, we're going to do exercises in bioinformatics field. So today, we're going to focus on 3 experiments. Maybe you still remember how we did the lab at part 1. So we collected cells with saline, and then we concentrate the cells, pour off most of the supernatant, and then we add only 30 microliter of the concentrated cell solution to 100 microliter Gel solution. And through boiling and spinning, we extracted DNA and purified it for the next step. So the next step we need to do is using polymerase chain reaction method to amplify that specific piece of DNA around that specific region we're interested to study with. Let me first introduce what is polymerase chain reaction using an animated movie.
Polymerase chain reaction, or PCR, enables researchers to produce millions of copies of a specific DNA sequence in approximately 1 to 2 hours. This automated process bypasses the need to use bacteria for amplifying DNA. Cycle 1. A PCR reaction starts with a denaturing step. Samples are heated to 94 to 96 degrees Celsius for 30 seconds or more to denature the target DNA. Next, the temperature is lowered to 50 to 65 degrees Celsius for 30 seconds or more. This allows the left and right primers to anneal to their complementary sequences. The primers are designed to bracket the DNA region to be amplified. The temperature is raised to 72 degrees Celsius for 30 seconds or more. This allows Taq polymerase to bind at each priming site and extend or synthesize a new DNA strand. Cycle 2. The temperature is raised to 94 to 96 degrees Celsius, denaturing the target DNA as in Cycle 1. As before, the temperature cools to 50 to 65 degrees Celsius, allowing more primers to anneal. The temperature is again raised to 72 degrees Celsius. Taq polymerase binds to each priming site and synthesizes a new DNA strand. Cycle 3. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So to do PCR for today's experiment, we need a DNA template, which is your DNA you purified. And we also need a lot of free nucleotides, A, T, C, Gs, that will help us to build up those new copies of DNA. And also we need a polymerase. Polymerase is important. It's in the name of this reaction. That polymerase will help to initiate the whole process. We also need primers. Primers are just like bookmarks. They will recognize and bind to that specific piece of DNA we want to amplify and help us to get the copies. Buffer is also important reagent for this reaction because buffer will provide the right pH for polymerase to work, as well as magnesium, which is a helper for polymerase. At the bottom of this slide, I listed the forward primer and the reverse primers sequence. So that's a primer we used. For each different polymerase chain reaction, we need different primer sequences to achieve the specificity of the amplification. So today we set up the program for 30 cycles. The denaturing step will be 94 Celsius degrees, 30 seconds each time. Annealing step, we will have 64 Celsius degree for 45 seconds. Extending step, we have 72 Celsius degree for 45 seconds. All right, so we needed 2 microliter of the DNA you purified and add into the PCR reaction tube to do this. Okay, I'm now going to show you how to do it with your hand. Okay, now I'm going to show you how to do the PCR step. To do the PCR, we need a primer mix here. All right, we need 23 microliter of that. So I will use the yellow micropipetter and I need to change the reading to 0.230. That's 23 microliter. And I need to press down to the first stop and then get into this tube, release my thumb all the way up, and there comes 23 microliter primer mix. You see it? All right, and then I'm going to add into this special PCR tube. This is called Ready to Go PCR beads tube, and it has a lot of things there. You can buy it from a commercial company. If you want, I can post the link there, and you open it up, and then you get this primer mix into it. Press your thumb down all the way to the second stop. Keep pressing back out. Then press this ejection button to change the tape. All right. Now you see in this PCR tube, I have this pink colored solution. That's everything you need for PCR reaction, but your DNA template. And where's your DNA template? Maybe you still remember last time we purified it. Okay, and we only need 2 microliter of that DNA, okay, solution. So we are going to use the green macropipetter, and we're going to change the reading to 0200 because the line here serves as a decimal point. So for 2 microliter, we only need 0200, okay, 2 microliter. And we're going to pick up the tape, right, and get your purified DNA. Remember, we use the adapter to when we do the spinning step. When we get it out, you can see this is 2 layers, right? At the bottom layer, is this better? Trying to focus it. Okay, so you see 2 at the bottom, that's the key legs, okay? And your DNA is at the very surface of the supernatant. So I'm going to press down to the first stop and touch the surface of the supernatant, then draw, and then come out 2 microliter. Can you see that? That's 2 microliter. This is better for you to see? Yeah, you can see, right? 2 microliter of the DNA solution. Add into this PCR tube. You touch the pink reagent, press down to the second stop, and then keep pressing back out, and then release your thumb. All right, that way you squeeze all the liquid into the reagent. Now you're ready to go for PCR. All right, now you see on the screen of this PCR machine, I have a program named PTC. So that's the program we're going to use here today. All right, so first of all, I'm going to open up the machine, and then I'm going to put this tube there. Now I'm going to put this tube into the machine like that, and then close the lid, tightening the lid. Okay, then I just press run. Now you know this is this program we set up. 94 degree for 30 seconds, 64 degree for 45 seconds, 72 degree for 45 seconds, and then repeat for 30 cycles, okay? And you start run, then the machine is going to start. All right, that's the PCR step. Very easy, right? Now, after the PCR reaction finished, we need to do DNA restriction analysis. So what is DNA restriction analysis? Let me use an animation to explain it to you now.
DNA restriction. Restriction enzymes are sequence specific. They recognize and bind to specific DNA sequences. EcoR1, for example, binds to the sequence GAATTC. Once they bind to their recognition sequence, restriction enzymes cut the sugar phosphate backbones of the DNA strands. EcoR1 is a restriction enzyme that cuts unevenly. leaving overhangs or sticky ends. The sticky ends can be reattached by another enzyme called ligase. It catalyzes the chemical reaction that rejoins the DNA sugar phosphate bonds.
All right, so we learned restriction enzyme is a special protein that will bind to the DNA chain and recognize specific sequence and make the cut. And different restriction enzyme recognize different sequence. It's like one lock, one key. And we have one special restriction enzyme called HAE3 that will recognize a sequence GG, CC, and then cut between G and C. That will help us to solve the problem here as how to distinguish non-taster and taster genes. Because you see here, the non-taster gene, they have, in the sequence, you have GGGC. Well, in the taster gene, you have GGCC. So the taster gene could be recognized and cut by HAE3 enzyme, and you got 2 pieces after digestion. Well, the non-taster DNA, non-taster gene, you have GGGC, and so this enzyme could not recognize and cut it. Therefore, after digestion, you still have 1 piece. Okay, then next step after we finish PCR, what we need to do is transfer 10 microliter of the PCR product into the new tube. And we're going to label this new tube with the letter U. U means uncut, undigested. Okay, then we're going to just add 1 microliter restriction enzyme, HAE3, into the tube labeled D, or the original PCR tubes. Okay, that means we're going to use the enzyme to digest it at 37 degrees for 30 minutes. Okay, I'm going to show you how to do this step now. All right, so I'm going to transfer 10 microliter of the PCR product into a new tube. To do that, I will use this green micropipetter again, and the maximum volume this one can measure transfer is 10 microliter. So I just go ahead, adjust the reading to 1000. That will be 10 microliter, okay? Now I got it, you can take a look. So that's 10 microliter, and I got the tape, okay? So this is the PCR. I finished the PCR, this is a PCR tube, and I just finished the reaction. I get it out of the machine, I press down my thumb to the first stop, get into the liquid, and then release it slowly. Now I get 10 microliter. You can see better, 10 microliter PCR product. And I got a new tube, and I put into the new tube and press down my thumb all the way to the second stop, and then keep pressing back out, and then release my thumb, and then change the tape. All right, then I close this tube, and I will label it uncut with a U there, right? U means uncut. Can you see it? Right, it's better see this way. Can you see the U I labeled on it? Yeah, that's it. Okay, then the other tube, I will put enzyme there. All right, so here is the enzyme I'm going to use to do the digestion here. It's HAE3, okay? I only need 1 microliter, so What's the number you're looking for here? Right, you only need 0, 1, 0, 0. Okay, I'm getting there. All right. Ta-da, that's 0, 1, 0, 0, 1 microliter. So I will get this tape. All right. And open up the tube. Press down to the first stop, make some press down to the first stop. Get into the enzyme tube and come out with 1 microliter enzyme. And I will add into the original tube, PCR tube here. Okay, press down to the second stop. Okay, and then come out, change the tape and close it. And I will label this with the letter D. D means digested, right? Okay, so how are we going to find the 37-degree incubator here? Yeah, we don't need it. We can just use the PCR machine and set up the temperature to 37 degrees for 30 minutes. That's what I'm going to do now. Now I set up a special program. I call it 37. So I put my 2 tubes there, one labeled U, one labeled D. U is the tube I didn't put any restriction enzyme, but 10 microliter of the PCR product. And I put them shoulder by shoulder here, and I'm going to incubate them at 37 degrees for 30 minutes. So I close the lid, tighten it, okay, and then I start to run this one. And you see, that's how I turn this PCR machine into an incubator for this step. When we finish this incubation time, and we're going to get both tubes out and proceed to the next step, to gel electrophoresis. What is gel electrophoresis? Now I'm going to show you another animation to help you understand.
Gel electrophoresis. Gel electrophoresis can be used to separate DNA fragments. Electrophoresis uses an electric current to separate different sized molecules in a porous sponge-like matrix. Smaller molecules move more easily through the gel pores than larger molecules. While at Cold Spring Harbor Laboratory, Phil Sharp, Joe Sandbrook, and Bill Sugden developed the DNA electrophoresis technique using an agarose gel made from highly purified seaweed. This could be used to separate DNA molecules, ranging from several hundred nucleotides in length to over 10,000 nucleotides. The gel is submersed in a tank filled with a salt solution that conducts electricity. Using a pipette, DNA samples are loaded into slots made in the agarose gel. The DNA samples are colorless, but researchers add a blue tracking dye. This makes it easier to load the samples and visually track the DNA migration through the gel. The phosphate groups in the DNA backbone carry negatively charged oxygens, giving a DNA molecule an overall negative charge. In an electric current, the negatively charged DNA moves towards the positive pole of the electrophoresis chamber. The DNA molecules move through the gel by reptation, a reptile-like snaking action through the pores of the agarose matrix. Smaller DNA fragments migrate faster and further over a given period of time than do larger fragments. This is how DNA fragments can be separated by size in an agarose gel. Sharp, Sandbrook, and Sugden introduced the use of the fluorescent dye, ethidium bromide, to stain DNA. Ethidium bromide binds tightly to the DNA double helix and glows when illuminated with ultraviolet light. This lets researchers see where the separated DNA fragments end up. A photo of the gel can be taken for later analysis. The size of any DNA fragment can be determined by comparing it to markers, DNA fragments of known sizes. A map of restriction enzyme sites can be generated by cutting a piece of DNA with different combinations of restriction enzymes.
Next, I'm going to show you how to load the gel. So when you load the sample, first line should be the letter, okay? So we usually load 5 microliter of the 100 BP DNA letter there. Okay, so we are going to use the smallest micropipetter and change the reading to 0500, okay? Right, so this is the 100 BP DNA letter, okay? I'm going to get first press down to the first stop, getting to the liquid, release. Now that's 5 microliter. Can you see that? Can you see it? 5 microliter. All right, then you get into the well, okay? Price down only to the first stop. Keep pricing back out, okay? Then you got 5 microliter. So the second thing you're going to load is your uncut, undigested sample, PCR product, okay? You labeled U on the side of the tube, right? Okay, so we're going to load 10 microliter of that into the gel. 10 microliter is 1000. All right, you price down to the first stop, you get all the samples out, and then you aim to that specific place, price down to the first stop. When you load the gel, you only price to the first stop, okay? The next sample should be the tube, from the tube you label the D, right? That's the digested sample. All right, and again, you load 10 microliter and put into the well. Okay, then that's all. You finish loading the gel, and then you just need to put the gel box together. This is the cover, and then you match the color black to this black, okay? Plug in to the power station, and I set up the power station V to this voltage 130 volt, and then press this button twice, and then you set up the timer by pressing the arrow here up or down to reach to 20 minutes. Then when you finish, you press the running figure here, then it start running. All right, that's how we run gel electrophoresis. Again, here we used 2% agarose gel, which means we use 2 gram agarose dissolved at 1x TBE buffer with the gel. We also run the gel at 1x TBE buffer. Okay, so next I'm going to show you how to get your result. As a reminder here, you can see if you are a taster, then your gene has GGCC in the sequence. It could be recognized and cut by HAE3 enzyme. Here is a picture from a previous experiment. 3 students participated here. Student number 1, you see under the D line, there's just 1 band. This means this student, in his genome, he only has non-taster gene sequence. So his genotype is little t, and we predict he is a non-taster as his phenotype. Student number 2 here, under the D line, you see 2 bands. That means this student, in his genome, he only has taster gene sequence. So his genotype is big T, big T. We predict he's a taster. The third student here has 3 binds under the D line. That means this student has both non-taster and taster genes in his sequence. So his genotype should be big T, little t, and we predict he is a taster. Okay, so that's all. We learned a lot today. We learned about PCR, we learned about restriction analysis, we learned about genotype-phenotype relationship. Next time, we're going to learn how to find out your PTC testing phenotype, and also we're going to move to computer sector to learn some bioinformatics. That's all for today. Okay, I hope to see you soon. Bye for now.